All right, everyone, it's 5:01. So welcome to all of you. I'm Dr. Ashley Kita, and I'm an assistant professor in residence in the Department of Head and Neck Surgery here at UCLA. I'm delighted to moderate this panel of experts today. So first, I'll be introducing our panel of experts, followed by a discussion. The Q&A function in Zoom is on at this time. So if you leave additional questions, we'll do our best to address them today, time permitting. So we have four wonderful UCLA experts joining our conversation as we discuss the intersection between engineering, neuroscience, and medicine, and how missing biological functionality can be restored through new advancements in technology and neuroengineering. All right, so we'll start by giving everyone the opportunity to introduce themselves. First, we have Professor Edgerton. He's a distinguished professor of neurobiology and member of the Brain Research Institute.
Oh, and thank you. And it's a very pleasant time to be here, and a lot of exciting things are happening at this time in the area of technology and physiology. I'm interested and have been studying for decades how we move. We know a lot about muscle cells, we know a lot about neurons, but we don't know how to match them well. We don't know how to fill that gap between knowing a lot about neurons or a lot about muscle and then being able to extend to behavior. So I'm considered an integrative biologist and physiologist, and my primary appointment now is in neurobiology. I've become interested several decades ago in how we move after paralysis, partial paralysis, and after even complete paralysis. And in that initial time, I learned how the spinal cord can control movement without any input from the brain. So that stimulated a central thought to me, which I'm still investigating today, is to what extent in different stages of paralysis or severities of paralysis, to what extent can we take advantage of the automaticity of our movement without any input or with minimal input from the brain. Now, this is one of the ways that our team is very different. We're focusing to a large extent on what is happening in the spinal cord, how the spinal cord actually controls movement. And that plus working at the systemic level or the integrative level is relatively unique, as opposed to there's a tendency to want to reduce the experiment to a simplest one so you can understand the details more, and it would be, so, for example, a cleaner experiment. It's very difficult to do integrative physiology. So you need to know almost everything that is happening in the body in order to interpret your results. And this is one of the exciting things that we have been working on for a number of years with Dr. Wentai Liu. We're developing a device that will enable us to collect information on multiple events that's occurring throughout the body in a real live performing person, and that or animal. And that device that's being built by Dr. Liu is one that we have planned in the future would be one that would be an implantable device for humans. So in this area, we have to have people with different expertise, and our team, and you'll see how different individuals that's on this panel will reflect different expertises, and that's really the unique feature that we can present.
Wonderful. Thank you for highlighting the collaborative atmosphere. So next, we actually have Professor Liu, who's a distinguished professor of bioengineering, electrical engineering, and is a member of the California Neurosystems Institute and Brain Research Institute.
Good afternoon. I'm Wentai Liu. So as Ashley said, I'm a professor in bioengineering, CNSI, electrical engineering, and Brain Research Institute. So my interest is in bioelectronics medicine, neuroengineering, and the biomimetic systems involving the central nerve system, spinal cord, and autonomic nerve systems. So my background actually is electrical engineer by training, and I have never worked on any bio-related project before 1988. Even I got my Ph.D. in Michigan, I was working on different topics, but sometimes the life could change dramatically because of unplanned opportunity. So this is the opportunity happened and my research career changed dramatically. So in 1988, I was a faculty at NC State University, but there are two eye surgeons from Duke University kind of tried to persuade me to work on a project or create a device to make the blind people to see again. And I didn't know anything about that, but I did not hesitate to sign on. So I learned a lot with the steep curve, the medicine and neuroscience terminology as an engineer. So I think I'm delighted to report that I am successful to overcome that difficulty. So I can speak fluently in medicine, biology, and engineering. So I we are people that can speak several languages together. So kind of very gradually the project and now that successful. So we do have a FDA approval device in February 2013. And that is the most happy moment in my life. So I think, so then a decade ago when I joined UCLA, and I was kind of recruited to create a Bionic engineering program sponsored by Dr. Patrick Swinshong. And thanks, Patrick. So I start collaborating with Dr. Edgerton on spinal cord implant by taking advantage of the technology that I have developed for the retinal implant. And the process is wonderful. The progress is really wonderful because Dr. Edgerton and myself and the other member, we form a very strong team that integrate the science and technology together. As Dr. Edgerton has explained, this is kind of unique features at UCLA. And I'm very humble, but very grateful that in my life, I do have the opportunity of making the blind people to see again, and also the paralysis to stand and even walk again. So an inspiration that we are familiar with the Bible story. Then few years ago, I started kind of wondering, beside the central nerve system, spinal cord, is there any other nerve system that I can tap in? And I find out that autonomic nerve system is a very fertile area. So I started working on that. And my goal is that I'm develop the method and try to understand the mechanism to regulate the immune system, the metabolism, and the secretion. So I think to my astonishing that I'm able to have some breakthrough discovered recently. And though I cannot tell, but I think I can assure you those are really a big breakthrough discovery and waiting for me to secure the IP or UCLA, and then I will be happy to share this result with everybody. So now I'm happy to be an academia, but also I'm enjoy to be an entrepreneurship. And I realized that this area do have potential. Not only we can help the people to reduce the suffering, we also have opportunity to solve some of societal problems. Thank you.
It's so inspiring to see you taking your technology to the people that it will help. And it's so wonderful to see how all of you are doing that. So next we have Professor Seidlitz, who's an assistant professor in the Department of Bioengineering.
Hi, so my work is really, so I'm a bioengineer by training, but I've always worked in the neuroscience area. And most of what I do is try to develop materials that can interact with the nervous system in ways that can control it. And so, for example, we try to design materials that can interact with cells in a way that leverages their native biology, right? So we know that they already have these intrinsic biological programs. So can we direct those to do something like regenerate the spinal cord after injury? And we do a lot of different things that we add to these materials to help sort of promote regeneration or direct cells to do what we want. But we work in the areas of both cell and gene-based therapies in addition to these scaffolds, so that the scaffold could actually be a carrier for a stem cell therapy or a gene therapy as well, so that those things can work together to really enhance regeneration. And I guess, you know, we work a lot, even though we're engineers, we work a lot from a biology perspective too, because I think as Dr. Edgerton sort of referred to, there's a lot that we don't really know about neuroscience, right? And so it's hard to, you know, come up with a way to really control that system without knowing everything about it, right? From an engineering perspective. So a lot of what we do is try to really design experiments that will give us both that biological information and advance our knowledge, and also can tell us, can we use this? Is this a good tool to actually spin off therapeutically, so that we can get something into a clinic or be used, you know, before we know everything about the system. So try and fold those two ideas together in what we do.
Yeah, it's wonderful to have you and talking about turning what we have into what we need it to be. So next we have Professor Samueli, who's a Chancellor Professor and Chair of the Bioengineering Department.
Hello, everyone. It's really a pleasure to be here. I was trained as a bioengineer. I did my Ph.D. at the UCSD, and then I started my faculty career at the UC Berkeley. And just 5 years ago, I moved to UCLA, joined the bioengineering department. My research is focused on cell and tissue engineering, and also trying to understand how biophysical factors can promote the tissue regeneration. So in the past, we have worked on various types of stem cells, including adult stem cells, placental stem cells, induced pluripotent stem cells, and embryonic stem cells. And we use these stem cells to derive different kinds of cells and for regenerative medicine applications. So in one area we're interested is the interface of nerve and muscle. So when we have nerve injury, you lose the communication between the nerve and muscle. In this case, two things we need to do. One is to accelerate the nerve growth. On the other hand, you want to reestablish the communication between nerve and muscle. So what we are trying to do is, one is to deliver stem cells to accelerate nerve growth. In addition, we're trying to address the interface between nerve and muscle by reestablish the neuromuscular junction using stem cell transplantation. So recently, we are also interested in biophysical therapies. So we know that between the nerve and muscle, the communication is through electrical pulse and chemical signaling. We understand that the biophysical factors, especially in this case, electrical stimulation, could play a very important role, and there's evidence that this could help the spinal cord regeneration. We are working on the peripheral nerve regeneration in this case. And the important step we are trying to move forward is to develop implantable, miniaturized wireless device, which could be used to stimulate the nerve. In this case, it could be a programmable stimulation. Of course, we just started research in this area. There are many things we need to do, and I will look forward to the discussion and share our research.
Yeah, it's wonderful to see all those things coming together to approach these problems from many different angles. So our first questions for the panel are centered around exploring the theme of where we currently are right now, so where the technology currently is. And so I kind of wanted to start that by asking just an open question of who are the patients that you think the technology you're developing will help? And so we can start it with anyone answering at this point.
Okay, I can add something here, I think, because after decades of experiments with animals, we have developed the implantable devices to the point that we are seeing significant gains in function. And the reason we're seeing significant gains in function that maybe others have not seen before, and including ourselves, is that we have been learning more about the physiology and the biology of the human, as well as, of course, the animals. But we've been able over the last 10 years to test many of the experiments with humans, and we've shown that individuals with complete paralysis for more than a year and some up to 20 years can learn to stand and can learn to take some steps. Also, their bladder and bowel function improves, and even sexual function. So this goes back to the point that I made earlier about the integrative biology, and that is every one of our organ systems are closely connected. So you can't affect one organ system. So we are trying to maximally take advantage of the integrative nature of our biology so we can improve multiple systems at the same time in the same individual. That's one of the advantages that we've seen, and we already know that we can have an important impact. So it's not something that's going to be 5 years or 10 years down the line. We already know that it's beginning to happen.
And then
So I might add. So Dr. Edgerton explained about the kind of potential benefit to those patients. But I think through the research with Reggie, we find out that a lot of autonomic dysfunction actually can be dealt with the spinal cord implant. So I think not only to restore the motor function, walking or standing up, but a lot of kind of autonomic function can be benefited by this kind of device.
Wonderful. And then for Professor Seidlitz or Professor Li, what are some of the challenges you guys are facing as you develop these technologies and some of the gaps in the technology we have?
So I can speak to that. So, the technologies that I am working on are more, looking at the level of how you control things from a cell and how those cells can really assemble into like a functional tissue. So it's kind of the flip side of looking at like the whole body organ systems at once, right? And so what we're really trying to do is can we just restore function, which, would be great, right? But can we take it even farther and really make new tissue? And really, so say you have, an injury where there's basically a giant cyst in the spinal cord, or you have a giant area of lost tissue in the brain, say after stroke or after some neurodegenerative disorder, can we actually replace that tissue and allow it to integrate back into the patient's actual tissue to restore function that way? So I think it's a little bit different approach in that way. And so I think I mentioned before, a lot of it is we need to figure out, how do we do that biologically? So how does that happen in development? Can, when you actually, say development in the womb, right? Development in young children, and can we basically mimic that and maybe even reactivate those sorts of programs in adults to really regenerate that. So we have to do a lot of work on, understanding the system and then really taking that information and learning how to control it. And there are a lot of things that are really important in that. I think a lot of people will talk about stem cell therapies, for example, and those have been in clinical trials for spinal cord injury. But the problem is that the way that it's been done in the past, they don't work widely in a number of patients. And even, in the patients that they do see some significant recovery in, it's pretty amazing how few of those cells are actually leading to that recovery, because something like 90 to 95% of those cells die immediately upon injection. So, one of our first things to really start tackling as regenerating a tissue is to figure out, what are the base things that we need to instigate these programs. And in adults, they may not have these cells that are really flexible enough to become new tissue. So if we can figure out which cell source, which kind of stem cell transplant works the best, and something that we can make to where, you know, an immune system of a patient is not going to reject it, and to where we can also basically scale it up, right? So that we can make a lot of it really consistently, which is hard to do with a living thing, right? So you can imagine, like in drug manufacturing, you're doing a chemical process, you can purify it later, but it's not changing constantly like a cell source is. So a lot of that challenge is really, you know, getting the biomanufacturing aspect in as well, so that we can do it in an affordable way that's predictable, so that you can really get it towards the clinic.
Great, thank you. And then I guess for Professor Lee, asking very similarly, like where's the technology lacking for you?
Yeah. So what we're trying to address really is this peripheral nerve injury. And this can be caused by many different things. For example, spinal cord injury, you also cause the peripheral nerve dysfunction. Or if you have a traumatic injury in different part of the body or organ or limb, that could be a problem. And in addition, when someone has a tumor removed by the doctors, part of the nerve can be injured as well. So for all of these cases, what I'm trying to do is to see whether a new technology could improve what we have now. Currently, really, for example, for the peripheral nerve injury, there are two things you can do. If you have a transaction injury, you use a nerve conduit to connect a broken nerve, right? And in addition, you want to do something at the downstream. Because when the nerve grows, if it's not fast enough, you have muscle atrophy downstream. And when you have extensive muscle loss, the muscle will not be receptive, even though the nerve can reach the muscle. So you will have a disability finally. So to address that, what we're trying to do, one is to accelerate the nerve growth, and two is to delay the muscle atrophy or promote the neuromuscular regeneration at the interface. So the challenge here for us is to find the right type of stem cells for this application. There are a lot of studies on stem cells for nerve muscle regeneration, but really it's not very successful. The optimal type of stem cells and the mechanism of regeneration still need more investigation. On the other hand, if you talk about biophysical therapies, if you use electrical stimulation, there are many things that need to be done, for example, by materials, right? It's the interface of device with the surrounding tissue. It could be biocompatibility issue, or if the material is biodegradable, you don't need to do a surgery to retrieve the device. And then you need to deal with degradation rate and regeneration rate as well. And if you have an electrical stimulation, in this case, it could be, for example, wireless stimulation or even ultrasound stimulation. And how can you achieve minimally invasive or non-invasive therapy? For example, if you have a device implanted around your nerve, you want to have 30 minutes treatment every day. And how can you do that without too much pain? You can do it at home instead of going to doctor's office. So those are the dream we hope to have in the future. But just the development of device and the integration of all of these things together by materials, device design, and even coding, signal processing, all of this, it could be a challenge. So a lot of work needs to be done in this area.
Fascinating. There's actually an interesting question from the audience very related to that, asking how the implants are powered. And I know you mentioned ultrasonography and some other kind of methods for communicating with the devices, but would they be battery powered? Would we talk to them through Bluetooth? Like what are you thinking, Professor Lee?
So if the question is about how we can achieve wireless stimulation, and I think there are a couple of things people are trying. Wireless is one obvious approach. Of course, there's some limitation regarding to the depth where you can put a device and whether this wireless signal can be transmitted efficiently down into the tissue. The other one is ongoing research is ultrasound stimulation, which could overcome this limitation if you can have a way to convert the mechanical energy from ultrasound into electrical signal to stimulate the nerve, that would be wonderful. So that's something we're trying to explore.
Wonderful. So then a question directed at Professor Edgerton and Professor Liu. So in my training, especially going through the emergency department, a number of neurosurgical services, training to be a head and neck surgeon, I saw a number of patients who had spinal cord injuries at kind of all phases of their recovery, from a tree trimmer who'd fallen, we saw in the emergency department, to people that we saw years down the line from a spinal cord injury. And so for patients with these sort of more chronic injuries, now several years out, what sort of recovery is possible with your technology at this time?
Well, most of our work has been with the chronic spinal cord injury in animals and humans. And so, and part of that's two reasons for that. Number one, in the first 6 to 12 months, there's often some spontaneous recovery, and you don't know if that's due to some intervention of yours or whatever it is. But after 12 months, it's generally thought throughout the clinical world that there's no further improvement that can occur after a year. So we began working with injuries later than a year and have studied them for years after, 20, even close to 30 years. So the system remains what we call plastic. And so we've been able to demonstrate these improved functions of multiple organ systems in a relatively short period of time, in a matter of weeks. And we don't know how much improvement they can occur because most of the experiments have been relatively short term. But the fact that we have thousands of people in our population that have chronic injuries, and because they have been injured for some period of time, is not a showstopper.
I think that's very encouraging to hear. And so for Professor Liu, unlike our other panels that discuss developing new devices for neuromodulation, your work, and along with Professor Edgerton's work, is focused on recovering functions using the body itself. And actually a component of all of our panelists' work does this. So why is that approach best, and how far are we away from recovering movement in paralyzed patients?
If we are talking about the restoration of the motor function, and there are about 3 approaches people are pursuing. Number one is the approach we are doing with Reggie. The other one is called BMI, the brain-machine interface. And the third one is that by the Mayo Clinic, they directly poke the electrode into the spinal cord to stimulate. So let's talk about the second approach, BMI. And this is a conventional field. It has been pursued by so many people, so many funding, and actually it gets to some of the kind of bottleneck. So as you see that the new company was formed and they pretty much adopt this kind of approach. So one of the problems is that, as Reggie pointed out, the automaticity is evolves through our evolution. So many years evolution give us that kind of solution. But now if we bypass this one, we directly harness the brain signal and decide the intention, then also we want to actuate or activate the particular muscle. But remember that the restoration of the motor function require a very sophisticated kinematic study. So any piece of muscle, when it's got to be stimulated, for how long, then what's next step, and what are the group we need to stimulate. Those are very complicated algorithm. So I believe most people right now, they say they want to use an artificial intelligence, but I think that's a kind of little bit hand-waving to kind of tackle this complicated phenomena we take it for granted as an evolution in the human being. So I believe that's why I believe UCL approach is much better than the BMI approach.
Let me, can I add one more thing to that? And again, going along the evolution idea, is that I often use as an example a dinosaur which weighs tons, and live for millions of years. And the size of their brain is about the size of a plum. And imagine that small brain controlling all of those functions that's necessary for many generations. And that kind of gives us an idea of how smart the spinal cord is. In other words, that's what we're trying to take advantage of. In some of the brain interfaces, some of the strategy is to bypass the spinal cord. And I say that that we are not as smart as 300 million years of evolution in trying to control these detailed movements. So we need to take advantage of the control, and the control, a major part of the control is in the spinal cord.
So I wanted to move our focus towards some of the things we're hoping to come about in this field in the future. So looking forward, and it's a question that I hope each of you will answer briefly in your own way, what do you foresee as the next imminent breakthrough in neuroengineering, and what technology needs to be developed to accomplish this? So kind of what's your holiday wish list for neuroengineering?
Let me answer first. So I wish that we have a device that has the capability of independently program a bunch of a set of stimulation parameters. This was because that on Reggie's research and my participation, we realized that even in spinal cord or different kind of vertebrae stage, you got to stimulate with different kind of either frequency, amplitude, or duration. So those kind of parameters need to be taken into consideration. When the person is standing up versus sit down, they all have a different kind of parameter. So if we want to restore the motor function, we got to take this into consideration. So the device must be smart enough that know what's the status, what's the current situation, then smartly issue the proper stimulation parameter. So I envision this device exists and cannot be big. You cannot give me a device like a car size. So we need a device maybe very small and very light, so the patient can be implanted and don't feel burden at all. This is my wishful list.
I guess I can say something related to that, but not exactly that, right? So if we can make these materials that basically, you know, the electrodes, right, that are interfacing with the system, and we can get some sort of closed loop system where it's really part of the nervous system, like Dr. Liu was talking about, right, where it senses what's happening in the body and it can feed back what it needs to, like the brain would do, right? I think one issue is that a lot of the materials we have for electronics right now that make really great electronics don't interface perfectly with the body, right? And so, you know, there are lots of movements towards making like soft electronics or making materials that can help interface those electrode type materials with the actual tissue in a way that, you know, they could stay there throughout a patient's lifetime. And it wouldn't be rejected, or there wouldn't be scar tissue that blocks off those connections of the electrode with the body at some point. So that's a really important area, I think, as well.
I think Wentai and Stephanie talk about this device is really something I'm interested in as well, to get a miniaturized, even biodegradable device that can be tailored in parallel to the tissue regeneration process, and so that you don't need to retrieve in the future. But I want to focus on stem cell therapy. So for stem cell therapy, indeed, a lot of things going on, but nothing has really gone through this pipeline to be approved yet. So there are some limitation of current stem cell therapy. In addition, really what we hope to have in the future is stem cells available off the shelf. This is the same issue as organ transplant, right? So when you have organ transplant, the patients have to take immunosuppressive drugs in the whole lifetime, unless they can establish immunotolerance. So for stem cell therapy, if you want to make them acceptable, not rejected by the patients, we have a couple of approaches. One is to probably build some universal stem cells that will be accepted by all the people. So in this case, you need to engineer the cells to remove some of those immunogenic antigens or molecules. And secondly, is to establish a cell bank that can be used for a certain population of people. So I think that will facilitate clinical application of the stem cell transplantation.
Yeah, and I could add to that a little bit and just say, I think this idea of having like a universal cell donor line, right, or a bank, maybe that some blood typed or however it might work, right, is probably more realistic in the short term than doing things like taking cells from the patient and taking them out and changing them. Simply just because of the cost, the risk with taking something out, keeping it sterile, putting it back in. But also, it's been really exciting to see all these technologies with, say, induced pluripotent stem cells, right? Where you can, which won the Nobel Prize a few years ago, but you can basically take someone's skin cells, right, and make them back into a stem cell and then turn them into a neural stem cell. And then you shouldn't have an immune response because it's the patient's own cells. I think the issue is that the technology for consistency between different patients is going to be really challenging. And so if we want, a consistent source that can get through, the FDA trials, which are very rigorous, then I really like this universal bank idea or universal donor idea, because you can do better quality control.
And from the clinical side, we don't always often have the lead time, sometimes especially with cancer cases, for something to grow in time for our need for it.
Yeah, and it would take, it could take a couple months really to get those stem cells to become what you want.
Yeah, and I did want to invite Professor Edgerton to say what's on his sort of dream list, if he is there. Otherwise, we'll kind of go. Oh, perfect. Did you want to say what you're hoping comes out of neuroengineering in the near future?
Well, I think that there's a lot of things going in the right direction, but we're not there. So I think the interesting thing today, compared to my experience in the past, is we have a lot of good ideas that need to be investigated. And when I say we, that is the area in general, because there's a lot of potential good ideas out there. So we need time. I often use the analogy, we're at the stage right now that's exciting, but it's still, we're like the Wright brothers' first flight. And so we are very optimistic with what we know now, and realizing what we need to know in terms of exactly how to stimulate or modulate the spinal cord and the brain to change its mood. That's basically what we're doing. We're not trying to induce a movement or a behavior. We're trying to modulate the state of our nervous system so it's in the mood that the person can voluntarily perform a given behavior. So that's where we want to go, and that's where I think we can get, and we can reach significant levels in the short term, but we should not even think about the possibility that we have reached where we really need to go.
Great. That's very important. So a question directed for Professor Liu. So all of our panelists have had success translating research to the clinical setting because they're thinking about these important problems. But I wanted to know specifically what your team and what you think academics in general knows that maybe large companies don't, that kind of has attributed to this success.
Well, thank you for asking this question. I need to be very careful to answer it without upset some of the players in the field. So I would say the major difference is the science knowledge. I think the neurophysiology knowledge exists at UCLA, particularly by Professor Edgerton. And our competitor in the big company, they usually take in the conventional, the BMI approach. And that's one of the fundamental difference. And I believe that, as Reggie said, I think we do have advantage. I strongly believe in that. Second thing is that those big companies usually have a deep pocket, they have a bigger resource, and they can hire 100 people. And we are university team, we usually are limited by budget and also the team member. So we need to play very smart. And I think let's go back to ask ourselves, what is the mission of university compared with the company? We do clearly see the difference. A university is set up to generate new knowledge and a new technology, then proliferate to keep the knowledge to the next generation. But the company doesn't have this mission. Company just say, okay, can using the existing technology and build device and succeed. So here we have a different mission. Therefore, we should preach our stronghold is a creativity. I think that's why if we are creative enough, we can do something better than company has done, even with a very limited budget. And so far, I think we are in the right direction. So we can compete with the company and we can develop a new design technology. So I believe that our technology is much ahead of our competitors.
Well, that segues nicely into kind of the next question of what sort of support it takes for us to reach some of these milestones that we're aiming towards. And so this is an open question for anyone who wants to kind of mention.
I think for the support, really, I say two ends. One is at the beginning, when you have some innovative idea, but you don't have any data or evidence to show that could work. Kind of a seed funding to support the research is very important, so that our lab or many others can start exploring the new direction. And on the other hand, I think that when we go through the development, in the process, probably we're going to need extensive multidisciplinary collaborations. So we need to reach a scale in that we have a team that include people with all different kind of expertise. We need engineers, neurologists, neurosurgeons, and all of these people have to work together closely to address the clinical problems, to develop technologies. I think, when we reach to a certain point, these interdisciplinary interactions will be very important. A center or institute type of organization will help to promote the collaboration and hopefully really translate the technology into clinical applications.
If I may, if I could add to that, I think that to do that, you need a lot of money because the way our granting system is, you can apply for grants and you always need to have someone else with certain expertise. But suppose you had a big pot of money, and that whenever you came up with a problem, you could go to the best in the world to recruit that person to help you solve that problem. You wouldn't have to wait a year or two before you write a grant and then finally get money. And by that time, everybody's lost their excitement about the idea. And I always use a comparison is really what we need to solve this big problem or these big problems with dysfunction we need the Manhattan Project. And that's exactly what the nation did. There was a very important problem to solve, and the smartest people, the best people were recruited to contribute to that. That's the strategy that we need to solve big problems like we're talking about. So that's not going to be done by NIH. There needs to be substantial sources from philanthropy, I think.
I wanted to ask for students watching, what advice would you give them in pursuing this type of work or in applying their degrees if they want to get involved?
I'll give it a shot because I switched my field from pure double E into biomedical. So I do face a lot of a steep learning curve. So I like to encourage students think big and open-minded. Don't just limit yourself to a particular field or a department. Looking for opportunity. If somebody hand you an opportunity, evaluate and take it. Like me, bravely taking that opportunity, and then I changed my career completely. So this field actually is very rewarding. If you work it right, not only you change people's life, you also see the entire family so kind of grateful to you. So that kind of reward is incredible as an engineer. Now we never get that kind of personal gratitude from the patient family. So we need to be open-minded and take the course, diversify, and especially for engineer students, I advise that you need to take some of the neuroscience or some of the biology course, and also really need to take a really good math. I really benefit by my strong capability of doing sophisticated math. So now I really get benefit. So I think not only I know technology, but I do have a mathematical skill. I can handle very sophisticated signal come out from the neural system.
Thank you. I did want to spend a moment looking at some of the kind of societal impacts and ethics of this technology. And so I had a question that I'd be interested in Professor Seidlitz's answer, which was, so should there be conversations about the ethics of human enhancement? I mean, particularly these stem cells we're talking about developing. And if so, who do you think should be involved in organizing and discussing some of those conversations?
Well, so I mean, I think there's sort of a difference between talking about like restoring a function that's lost that, say, you know, maybe the majority of the population has, right, or it would be desired, and actually like providing like an enhancement beyond that, right, to give some sort of super capability, right? So I think there's definitely a line there. You know, stem cells, it's sort of an interesting it's sort of an interesting thing, right? Because now we really have sources of cells that are either banked, so you don't have to take them from, you know, an embryo or something like that, right? Or we could use these sort of induced pluripotent stem cells where you could take them from the skin, and maybe you could even make your engineered line or your universal donor line out of that. And you know, it's not really any harm to the person that it comes from initially, right? So a lot of the ethical issues that I think were very concerning at the beginning of particularly stem cell technologies and when they were first sort of brought into mainstream research, people have mitigated a lot of those. You know, I think there are, I have always appreciated, as far as the people who are really doing this, right? So we'll often, you know, at conferences like the Biomedical Engineering Society conferences, you know, there will be sessions and things where the experts that are in that area can sort of have these discussions and start thinking. But I from what I've found is the most helpful guidance and these sorts of things that I've seen is when you actually get panels put together at the national level. So through NIH or this congressional panels that can really study this and write up a document and provide guidance. So for example, in using gene therapies, right? So introducing different DNA, you know, that when that started, that was incredibly controversial. There was a panel that actually brought together all the scientists, brought together bioethicists, brought together clinicians, and everyone was able to sit down and give guidance, you know, to the community that we could really follow. And I find that that's very helpful whenever any of these sort of new things come up, and we have to really consider, you know, what is the larger implication of pursuing this research.
We did have a question from the audience I wanted to bring up that for Professor Edgerton and Professor Liu. So regarding the complexity of the spinal cord and evolution, do you not think that the combination of more complex models and neuroplasticity could evolve to generate accurate signals?
I'm not sure how to answer that question. I think the way to look at the nervous system is not to see a difference between the brain and the spinal cord. they're all connected and all completely interdependent. And for some reason, the idea has developed that everything is happening in the brain, but almost everything that's going on in the brain, the only way to communicate it, except for the new brain interfaces, has been movement. Every bit of your behavior in some way or another is controlled, is communicated through movement. And so I think that the what happens in the brain is really very similar to what happens in the spinal cord in terms of communication and the basic mechanisms. So I think it would be hard to develop smarter communication systems that is already in the brain and the spinal cord. We need to learn how they're working, and then we can take advantage of how they're working. But we know so little about how these neurons work as a system. For example, if you think of one neuron, any neuron in the body is ignorant of all of the information it receives. It doesn't know the meaning of it because it receives information from so many sources. And then even where it projects to another neuron, it doesn't know what the impact is going to be on one neuron because that one neuron is constantly receiving different signals. Where the mood of these networks is constantly changing throughout our life. So we need to know more about what's there rather than try to improve on what's there.
And I guess that I wanted to bring up the point also of the, you know, where you get the funding to do this, where you get the big initiatives. So that we were kind of talking about as an analogy as like the Manhattan Project. I do think that the brain mapping project that was started during the Obama administration has given us incredible amounts of information about how these things are interconnected. You know, we still have farther to go, but we never would have gotten there without that large influx of support directed at exactly that area. And, you know, once we have that information, now we can try to figure out how to use it.
Wonderful, thank you. So I'm going to ask you each for final remarks in just a moment, but I also wanted to say that we received several questions about repairing vision and memory. So I briefly wanted to mention that this will be discussed during the next panel session on January 27th. We'll put up a link after the final remarks. But going through and asking each of you to kind of share your final thoughts as we're near the end of our hour together and before we wrap up. So first, Professor Edgerton, is there anything you'd like to share?
Well, I'd like to reiterate, I think if we could get the engineering and the biological community together that's interested in brain tissue interfaces, we would be thinking of the brain and the spinal cord as a single organ and trying to take advantage of how that interaction can be applied to begin to have greater levels of recovery than is possible at this time. So I think the development of the brain interface that's a great technology that I think is really going to make a big difference. But I think we need to have more. I think what has happened is that the things happening in technology have been going so fast and so impressive, but the physiology has not advanced as well. And because the type of physiology at the systems level is no longer popular. It's what happens at the cell or the gene. And knowing what happens at the cell or the gene, there's nothing there that could tell us that we would have observed these phenomenon so that we could have recovered these functions. So there needs to be systems to cells to synapses. That needs to be in the team as well as every engineering. And that's why That's why we need so much money to take advantage of the real creativity that's possible.
And Professor Liu, do you have any remarks you'd like to share?
Yes, I think I second Dr. Edgerton's opinion, and I believe that usually we are doing very good, but I think we can be even better, not to encourage the South Campus medicine campus and the engineering, that we have a more kind of openly dialogue, and so make sure that we're not get embarrassing by talk to doctor or engineer, or doctor talk to engineer to be scared with the Fourier transform and so on, the kind of mathematics tool. So second thing is that I really think about that we need to kind of revise our curriculum to encourage this kind of interdisciplinary or multidisciplinary research. And currently, we do have grassroots effort, but I really hoping the leadership can see this one and take some correct measure to encourage that. So third thing is that we need to encourage the student and even the faculty to do entrepreneurship, because that's the way can make a progress of this new technology to become available for the unfortunate human patient. And fourth one we said and touch in this panel is the FDA play a very important role. So we need to have university faculty to really understand the role of the FDA, because that we have a very good design, but you cannot pass the FDA's approval, then it's useless.
Yeah, and one of the big advantages of UCLA, I thought it when I was training here before faculty, was the proximity of all of these different things to each other and the potential to collaborate and communicate. Professor Seidlitz, I wanted to ask you next.
So yeah, so I was also going to mention something about how, you know, this type of work is incredibly interdisciplinary, right? So you really have to coordinate, you know, the people working with the patient's needs, you know, really identify what is needed. You may have some really great idea, right? But if nobody's going to use it and it's not practical, you know, along all these lines of, can you manufacture it? Can a physician use it easily? Is it comfortable for a patient? Then things are never really going to get translated. And I think that's one thing that it's been really nice for me about being trained in bioengineering departments, which is, you know, I've learned to speak all of those languages, right? And how to communicate with people at all of those levels. And I think that's really important, you know, for students to not be scared of learning neuroscience and learning physics, you know, and learning electrical engineering, learning about circuits. I mean, all of those things you have to kind of dip your toes into and get enough understanding to put these big ideas together. And I think, you know, one way that we can really start to do that is by building centers, you know, with a specific purpose, right? Where we get all the people that are experts in those little areas, but know just enough about the other areas that, you know, they can be glued together to really push these things forward, so that we're not all kind of working separately, right? You get a whole bunch of these labs together that are usually very small and make a bigger coordinated effort.
Yeah, that's a wonderful argument for centers of excellence and making these teams. Professor Lee.
Yeah, so in the Q&A, there are some questions about the research and career development in this area. I think I can make some points here. Neuroengineering is a really broad area. It has so much space to allow every, you know, for people with different background to participate and contribute to the development of technologies in therapies. So for example, in bioengineering, if you talk about molecular, cellular, and tissue engineering, you know, stem cell delivery or drug delivery, or can also be used to facilitate regeneration process. If you talk about biomedical devices, it's obvious electrical stimulation or even some monitoring device, wearable devices. Those are all important areas. If you talk about imaging, you know, we didn't get into that today. For example, brain imaging or imaging to monitor the nerve growth. Those will be new areas to get into. Or if you talk about data sciences, right? So the signaling, all of these signals from the brain or spinal cord and used to control the nerve function. And a lot of data it needs to be processed and understood. So all of these are possibilities. And more broadly, you can think about electrical engineers, mechanical engineers for prosthetic design, and etc. So just so many things we can do. And I will encourage all of the trainees and students look into this area.
Those are all great points. So thank you so much to our really lovely panelists and everyone in our community who joined us today and submitted such great questions. Everyone who registered today will receive an email with video links and a survey from today's event. We encourage you to complete the survey and provide your feedback on your experience. If you have any questions or would like to learn more, please contact Samantha Lang in medicine or Laura Yu in engineering. And lastly, we hope you join us for part 3 of the series, The Future of Prosthetics, Memory, Vision, and Communication on January 27th. Thank you all so much, and please stay safe and take good care.